computer. Very good. Well, it's 5 o'clock Cape Town time. I think we should get started. So we're very happy to have a friend and collaborator of mine here to talk today, Associate Professor Ryan Smith, who is an Associate Professor at the Laureate Institute for Brain Research, has a very interesting background in philosophy and psychology and neuroscience, does an enormous amount of work. I hadn't actually looked at your Google Scholar for a little while, and there's well over 150 papers on there, which is intimidating, particularly given that they all seem to be some seriously good research. So we're extremely happy to have Ryan here to talk today, to tell us about novel approaches for understanding the neurocomputational basis of interoception and emotion-cognition interactions. Over to you.
Thanks so much, Jonathan. And really appreciate the chance to be here and give a talk to you guys. Certainly really enjoyed getting to come out last fall to visit Cape Town. It was very, very fun. So jealous in some ways of how pretty your city is. But yeah, just to get started, I kind of packed a lot in here, so I'll see if I can see how much of it I can get through. I also know there's fairly varied backgrounds here, so I'm assuming that most people in the audience are more kind of on the computational end than they are on the kind of psychology or neuroscience end, but we can obviously talk, anybody's more than welcome to ask questions. If you want me to get through everything, we might want to take questions at the end, but I mean, again, it's kind of up to you guys. But the main sort of overarching focus of what I'll be talking about is some recent approaches that we've been using to try to understand neural and computational mechanisms associated with interoception and emotion-cognition interactions. So I mainly do what's called computational psychiatry. So that means that we take computational modeling approaches, we build specific computational models of behavioral tasks, and then we ask either healthy individuals or patient populations to perform those behavioral tasks, sometimes while also doing neuroscientific recording like fMRI or EEG or different brain imaging methods. And then we'll try to fit those models to people's behavior and use that to try to infer differences in underlying computational and neural mechanisms. So here I'll be talking about that in relation to some work we've done on interoception. So first, I kind of have to describe what interoception is. So interoception is basically perception of what's going on inside of your body, which is thought in many ways to be important to emotional experience. So perceiving, my heart rate, perceiving how difficult it is to breathe, perceiving, kind of feelings in my stomach, things like that. So, did I just feel my heart rate? Do I feel hungry? Am I getting enough air to breathe? Things like that. But importantly, it doesn't always need to be conscious. Much of the time, the brain is keeping track of stuff going on in your body without you noticing it all. Usually, it's only when something's going wrong, actually, that you start to be really aware of a lot of things going on inside your body. And some things we never do. So, for instance, we don't perceive blood pressure, but different parts of our brain are definitely keeping track of it and regulating it, like in real time. Also, at higher levels, interoception often requires interpretation. So what does this bad feeling in my stomach mean? Am I hungry? Does it mean I'm nervous? Does it mean I'm sick? Right? There is this kind of higher level inferential component. And usually, and again, I won't spend much time on this because of the audience here, but the way that we model interoception is the same way that we model any type of perception in neuroscience these days for the most part, which is based on combining some sort of sensory input with some prior expectation using Bayes' theorem. So we just have some prior expectation about what is going on in our body. We have some sensory signal, and we have a likelihood function that says what body state is most consistent with that signal by itself. And then we use that, we combine those things to infer some new posterior updated belief about sort of our best guess about what's going on inside of our body. And usually the way it's discussed is that the assumption is that whatever we consciously perceive is something like the mode of that distribution. So like the brain's best guess at a given moment about what's going on inside the body for a given signal. Now, again, I won't go into this in much detail here, but what's the reason why the brain would have to be doing something Bayesian? Well, basically, it's because sensory input is noisy and it's always by itself consistent with multiple interpretations. Now, you can kind of see this with example where a really big leaf far away or a small leaf up close, both produce identical images on the retina. So your brain really has to have a model saying, are big leaves far away more likely than small leaves up close? And that's what it has to use to kind of determine what the more reliable guess would be. Again, very often not conscious, and you can make this point in some more fun ways with like visual illusions, where here, for example, most people, when they look at this square labeled B and this square labeled A, have a very strong perception that those are different colors. But if right, if I actually draw a bar down through them, they're actually the exact same luminance, exact same color, or the exact same light is emanating from them. But because your brain has this kind of unconscious prior that under a shadow, the same light would be emanating from, right, a different underlying color, then you perceive it differently. So that just kind of is meant to make the point that this is not conscious prior expectations I'm talking about here necessarily, just the fact that under the hood, the brain has priors and those influence what we perceive, whether or not we know about it. So the idea is that the same thing is happening with interoception and interoceptive computation. So signals that the brain receives from the body fluctuate over time. They're noisy. It's not always clear what they mean. They often, or really always, probably admit to alternative interpretations. So the brain needs to use prior expectations and continue to update those prior expectations in relation to the current context about the best way to interpret those signals. So things like, okay, did I just exercise? How long has it been since I ate? And there's vulnerabilities that come along with that, just like in any Bayesian system, where if we trust prior beliefs too much, if they're overly precise, then we might just perceive what we expect to perceive. If we don't trust the signals enough, instead we might arrive at really kind of noisy or implausible, unstable interpretations. So things like, I must be having a heart attack after feeling just your heart skip one beat. You can do this kind of like essentially noise fitting. You could think about it that way. And we also need to learn appropriately after unexpected input. So we need to be able to change our prior expectations over time based on the patterns in past experience. So overly precise expectations, insufficiently precise expectations, problems with learning to update those expectations, all of those things have the chance to lead to different sorts of emotional, or could underlie, I should say, different sorts of emotional pathology or problems regulating bodily responses that are associated with emotional or other psychopathological issues that arise, such as exaggerated or prolonged responses to stress. For example, if we can't keep track of what's going on in our body, we also will have a difficult time regulating it, or that's the assumption. So interoceptive dysfunction does look so preliminarily like it. There's some evidence for it in different psychiatric conditions, including depression, panic disorder, substance use disorders, and eating disorders in different ways. But the literature on this is fairly, it's not entirely consistent, and there's not that much of it yet. So it's a very ongoing area of work. But in this prior work that's been done, computational mechanisms have not really been investigated, or not prior to those sort of things at all. I'll share with you. And this has mainly been done in the context of cardiac interoception, so perceiving heartbeats. And so other interoceptive channels also hadn't really received much attention for a number of reasons. So the main thing that I'll focus on today is three different kind of lines of research that we've been doing, focused on three different interoceptive channels, and the sorts of computational modeling and behavioral approaches we've been using to try to study them. So first is gastrointestinal perception. So the way we sort of perceive and make sense of signals coming from our gut, so our stomach in particular in this case. And then cardiac perception, as I mentioned, so this would be perception of heartbeats. And then finally, some work on respiration that we've been doing. And in this case, it'll be slightly different because we're trying to make a stronger link between interoception and emotion in that case. So it's a respiratory induced emotion paradigm, and how that influences decision making. So the idea here is that, building on previous work, can computational modeling in these areas offer any novel insights, both in terms of basic science as well as in terms of potential clinical relevance. So part one, gastrointestinal interoception. Like I said, basically no work had been done on the computational basis of gastrointestinal interoception at the point we've been doing this. So it's a very kind of novel area and exploratory at this point. But we think that effectively perceiving and regulating what's going on in our stomach and our gastrointestinal system more broadly is probably very important for a number of psychiatric and psychosomatic conditions, such as eating disorders in particular, which we've been looking at most recently. So for this study, or set of studies, participants completed a GI perception task, and then we used a computational model of behavior on that task to try to differentiate a few different computational mechanisms. So one was essentially the precision of the likelihood function, so the trust in the sensory signal, or what we're just going to call the interoceptive signal precision estimate. We also measured a prior bias, so essentially the bias in the prior expectation of what you're going to feel. And then also aspects of learning, so how quickly prior beliefs changed over time, both in terms of increasing expectations and then how quickly those expectations kind of decay away. So we can call those learning and forgetting rates. And this was in 40, the initial study here was in 40 healthy volunteers, so we're not looking at a clinical group just yet. And if people want to see, this work is published in this paper here, and we also have an updated paper in a larger sample size that came out more recently. So those are places to look if people want more details. But the way that the task works is it's kind of an interesting task. So there's this company that makes a capsule that you can swallow, and this capsule has a little mechanical motor inside of it. And you can pre-program it to vibrate at specific frequencies and specific times and specific intensities. You pre-program it right before people swallow it, and then you have this sequence of vibrations that happen, and we know what the timings of those were, and then we just have people push a button every time they feel a vibration coming from their stomach. So we have a kind of experimentally controllable signal coming up from the stomach to the brain that we can relate to people's perceptions based on the button presses. And the way that we have it programmed, for what I'll show you at least, there's different ways of doing it, but in this we just have a pseudo-random vibration sequence. It's over a 30-minute period, because that's about how long these capsules stay in your stomach. And there were 2 different vibration strengths to try to manipulate the likelihood effectively. And so we just call those strong or weak. And while they were doing that, we were also recording electroencephalography, so electrical activity in the brain through the scalp, and then other peripheral physiological measures as well, like heart rate, skin conductance, and things like that. So the way the model worked, and I'll walk you through this in a little bit of detail now. This will also be reused in very similar form for the cardiac perception stuff, but the way the model works, it looks a little complicated here, but it's just a set of vectors and matrices for implementing a discrete version of Bayes' theorem. So there's just the actual observation itself that comes in, so either a no vibration or a vibration, and then we have a likelihood function here, a likelihood matrix with a parameter IP, so interoceptive precision. It just controls how precise or blurry this likelihood function is. And then we also have, and so that corresponds to essentially the precision of the sensory signal in our version of Bayes' theorem up here on the right. Then we also have another matrix that represents that bias and prior expectations. So PV here is essentially the bias in the prior that a vibration is going to be felt at a given time in the trial. And so we also fit that to participant data, and those get combined together. And so on every given trial, the person has a posterior distribution over the possibility that they either did or did not feel a vibration. So like I said, it's a fairly simple model. And so from that, on a given trial, it's assumed that the brain is inferring some distribution over the likelihood, so the probability that a vibration was actually occurred. And then there's also a learning component to this, and this works based on the fact that this model incorporates a sort of directionally categorical model aspect to it, which basically means that the beliefs about or at least in the model that wins, which I'll talk about again, the values for PV start at a particular value which we fit, but then they update over time based on accumulating scaled counts in a directionally distribution. And that's just described here. Again, we can go through that in more detail if people want. And that's modulated, and each of those updates are modulated by a learning rate parameter here. But to give you a sense, basically you could think about it like this, is that at a given time, say there's a vibration, someone's prior expectation that they're going to feel a vibration again goes up, goes up, goes up, and then it goes down when there's a delay for a little while, and then it goes up, goes up, goes up again. And then goes down once a vibration doesn't happen. But by messing with the values for learning rates and for forgetting, which is essentially just the value for this eta here on trials where a vibration was not presented. So by making those asymmetric, you can also end up in a situation like this, where prior expectations build quickly, but they decay slowly. So over time, the prior expectations on average end up being pretty high. And so there ends up being this prior bias. So the idea is, can we find individual differences in that, as well as neurocorrelates? Yeah.
Can I ask you a quick question here?
Sure.
So just naively, I would think, so the thing vibrates and it vibrates for a little bit of time. And so there is an expectation that over that time, it will continue and then it stops. Should the expectation not go up, the sort of anticipatory prior, sort of believing more and more that it's going to happen any moment?
Well, so I mean, I think you can kind of think about this in terms of like an expectation about like the rate at which these vibrations are going to occur.
Mm-hmm.
So these are a little kind of discrete,
like
it vibrates for a second, it stops, they push a button if they felt it, it vibrates again, it stops, they push again if they felt it, right? So we actually more kind of control the frequency that the vibrations are presented. Like each vibration is very short.
Okay. And so if they haven't seen it for 20 seconds, the belief that it will happen in the 21st second is fairly low because they've only seen it once over the last 20 seconds.
Exactly.
Okay. Yeah, thanks.
Okay. So anyway, so that's more or less how the model works. And in this particular class of models, this is based on some work by Carl Friston that's extended this sort of approach to also include decision making. You can actually use these models with a variational updating approach to I mean, it's more detailed than I'll go into right now, but the point I want to make is just there are ways in which you can use these updates to simulate event-related potential magnitudes based on the rate of change in the belief update after each observation. And so we can use that to make predictions about how strong people's neural responses would be if we measured them with EEG during the task. And if people want to know more about that, there's a tutorial that we've put out, as well as a fairly recent review, not that recent anymore, of way these sorts of things have been used more generally in other areas of clinical pathology and psychiatry. But the neural hypotheses we had primarily with EEG was that people who would have higher interoceptive precision values, so higher IP values that we would estimate based on behavior, that they would also show stronger event-related potentials in the brain because they would be more sensitive to the signal itself, essentially. And where in contrast, people who had stronger prior expectations would show weaker responses because there would be less belief updating because the signal would be more sort of already expected effectively. And then more importantly, that belief updating rates, right? So the learning rates and the forgetting rates, that those should modulate these responses in a way that kind of depends over time on the way in which they change the trial by trial influence of prior expectations. So we would expect faster learning to lead to stronger expectations on average and therefore weaker neural responses, and faster forgetting would lead to weaker expectations on average and therefore stronger responses. And we would expect that to be the case, especially in frontal regions that are implicated in these top-down prior influences. Just kind of in brief, we fit a bunch of different possible models that included some or all of these parameters or other potential parameters and did Bayesian model comparison. The model that won is this model 6 here, and that includes the interoceptive precision. It also includes a change in interoceptive precision when it switches from the strong to the weak vibration strength. It also includes the initial prior belief, as well as learning rates and the forgetting rates that I mentioned before. So in terms of results, some of this doesn't matter in too much detail, but on the left here, what you'll see is just a correlation matrix across all of the EEG electrodes. And if you do a latent factor sort of dimension reduction thing on it, what you find is that there's basically just one latent factor that corresponds to the back of the brain, essentially, and another one that corresponds to the front of the brain. And that's true both at an early time window and at a later time window. So what we did is we just extracted factor scores for those two latent factors across electrodes and used those as our neural responses just to keep the dimensions from getting too high. And so doing that combined with a parametric empirical Bayesian approach to looking at relationships between the model parameters and those EEG responses, the results we find, so I'll show them separately for the parieto-occipital, so the posterior, the back part of the brain responses, and then the front part of the brain responses separately. So for the back part of the brain, so the latent parieto-occipital factor, we see pretty much what we would expect. So that both in the early and the late time window, higher values, people with higher interoceptive precision values also show stronger ERPs. So importantly, this kind of back of the brain that I'm talking about is more associated with initial sensory processing. So that's more or less what you'd expect. People with higher sensory precision estimates are going to be the ones that have the stronger belief updates in sensory areas and therefore should have stronger measured neural responses. Also, that's weaker in cases for people where that precision drops more when it goes from the strong to the weak vibration strength. So that's just kind of showing that the strength of the vibration also mattered, as well as you can kind of think of it indirectly as the kind of the steepness of the drop in people's subjective precision when the signal changes, the signal strength changes, it also modulates these factors. And if you just look at the correlations with each of the different electrode responses, you can kind of get a sense of the strength of these relationships. These are just each of the correlations themselves. And it's primarily, you can see, two sensory precision related parameters that are tracking these posterior brain responses. In contrast, if we look at the frontal responses, at the early time window, we also see an effect of the signal itself, which would be kind of like a fast, early kind of feed forward sort of response. At the later time window, though, when we expect the kind of prior influences to really be kicking in and the learning to be happening, we instead primarily see significant effects of the forgetting rate, so the decay, how quickly decay in prior beliefs happens over time. And so here you can see that those are fairly strong, and they're in the direction that we would expect, right? So people who forget more quickly should have less biased prior expectations on average, and therefore their neural responses associated with belief updating should be higher. And so just to kind of give you a little bit of a visual sense of what this looks like, this is what the ERP looks like coming from the back of the brain, so this POZ area back here. And that's what the correlation looks like between the precision estimates and the neural response strengths. You can see it's a pretty nice correlation, pretty well spread. And instead, if you look at the frontal responses, so this FZ electrode here, just as an example, it's in the opposite direction. And if we look, and this is what the correlation looks like between that and this prior decay rate, so this learning rate in the absence of vibrations. So again, both in the expected direction, significant, and kind of just validate the fact that the model seems to be tracking patterns in behavior that are also relevant to individual differences in brain responses on average. Also, just as kind of for validation, People did report higher self-reported confidence during the task if they had greater IP estimates, as well as less self-reported difficulty. They also showed stronger skin conductance responses, as might be expected if there's high surprise, right?
Sorry,
So
can
yeah,
you just go back? So self-reported confidence, does that mean sort of self-confidence in terms of how confident they're feeling, or what does that mean?
no, it's just kind of like after they do the task, like how confident they were that they performed well.
Oh, I see. Okay, sure.
And then same thing, like how difficult they found the task for self-reported difficulty. Whereas this one is an actual physiological measure, right? So where greater skin conductance corresponds to greater autonomic responses, which can be thought of in terms of a kind of measure of unexpectedness or surprise, or salience. Maybe it would be a broader way to talk about it. So as a kind of summary for part one, then it does look like there's a greater kind of trust in the sensory signal in some people than others, and that does amplify neural responses, whereas prior expectations look like they attenuate those responses, and that they are modulated as expected, in particular by faster decay rates in prior beliefs. So this opens up the possibility to study abnormalities in GI computation and psychiatric disorders. And we're currently collaborating on a study doing disorders, and the results of that will actually be presented in a couple of months at the Computational Psychiatry Conference in Minneapolis. Long story short, the main thing that looks like it differs in eating disorders is that they show stronger prior biases against feeling vibrations, and also that there's a learning rate asymmetry that kind of maintains that difference, maintains that kind of bias against feeling things. So anyway, it's interesting. If anyone's going to come to the conference, then you stay tuned. Okay, so part two then. So on cardiac perception, this is a work where our group is more kind of explicitly shown results in patient populations as well. So there's three different kind of papers here that people can look at. Some of the first one here is the main one I'll talk about, but these other two more or less just replicate the same results in new samples. But the idea here is you can think about it as kind of structurally similar to the GI task. But here, what's happening is we're just having people pay attention to their heartbeat, and then they just tap the button each time they think they felt their heartbeat. So pretty simple. But we manipulate a few different we manipulate the way they do that in a few different ways that are kind of designed specifically to target either the prior expectation component or the sensory precision component. So there's three kind of 1 minute conditions where we have them do that. There's a baseline condition where we just say no, where the instruction is no guessing. And basically that just means we tell them, don't press the button unless you are very confident that you actually felt your heartbeat. Whereas in another condition, we say, it's okay to guess. So basically just, push the button whenever, you think your heart probably beat. So this no guessing one really turns down, right, the influence of prior expectations about heart rate. Whereas in this guessing condition, people are more or less just tapping when they think their heart probably beat based on their prior expectations about heart rate. So you can think about this difference in instruction as altering the top-down kind of prior bias influence in particular. Also, we have a third condition where it's still the no guessing, like this baseline, but then we also add a breath hold. So people hold their breath during the period of time where they're doing the tapping. And that's a way of kind of non-invasively just increasing the strength of the actual heartbeat signal. You could try it if you want, hold your breath, and then start paying attention to your heartbeat, and you should notice that it will feel stronger. So that's a way of specifically altering the signal strength or the sensory precision in the jargon we're using.
Can I ask a question there?
Yeah.
In the no guessing and the guessing without the breath hold, do you study how they're breathing? Because presumably some people would sort of naturally modulate their breathing to try to feel it more strongly.
I mean, I think that I wouldn't expect there would be a difference between the no guessing and the. Are you saying you think people would breathe differently in the guessing and no guessing conditions?
That they the breath hold might actually be something that they naturally do to a certain extent in the other conditions as well.
Oh, no, yeah. I mean, we're constantly monitoring their respiration. No
Okay.
one's holding their breath during
Okay.
the other conditions. Yeah. It's an interesting thought. Okay, so anyway, you'll recognize this, basically the same computational model. It's just the heartbeats instead of the vibrations going in as the observations, it's the actual EKG trace that's going in as the observations. We still have an IP parameter and a prior bias parameter, which is calling it PHB in this case. There is one other difference, and that is that this raw IP signal here actually will be high, or I should say it will be far from 0.5. It will be precise either toward 0 or toward 1 if people reliably tap either right before their heartbeat or right after because of the way the time windows are divided. And so minimal precision here really is like 0.5, right? It's basically saying that the way they tap is kind of unrelated to the dynamics in the heartbeats. And so what we do in this case is we just take that initial kind of IP value, we subtract 0.5, and we take the absolute value of it. And then we can use this raw IP value also as like a kind of separate estimate of whether people are more kind of like anticipatory or reactive. But the idea is that the closer to either 0 or 1 here, the more precisely the taps are actually tracking the pattern in the heartbeats. Okay, so in this, this is a much larger study. So here we had 52 healthy comparisons. We also had 15 people with just an anxiety disorder, 69 with just depression, 153 with comorbid depression and anxiety. We also had 14 eating disorders patients and 131 with heterogeneous comorbid substance use disorders, primarily methamphetamine use disorders and second most opioid use disorders. So in terms of model comparison, another thing that's different between this model and the GI model is that model comparison favored models that only had IP and PHB, so they didn't suggest any within condition learning. So that suggests that expectations, at least within each condition, did just remain stable within over time. So we only compared those two parameters between groups. The main finding of interest is here. And what this is showing is that for everybody, so if you look at the guessing and the no guessing conditions, so this kind of salmon thing and this green thing, pretty much none of the different groups differ. But if you look at the effective breath hold, there's this fairly large effect size jump in the interoceptive precision in healthy participants, whereas that change is not present in any of the clinical groups. So there you can see that really it's something about the difference in the way that changing the signal itself is able to update precision estimates in healthy participants, whereas in the clinical groups, this kind of precision estimate may be inflexible to the actual signal itself.
Can I ask here, so has this been, what happens if you stratify over sex, given that the distributions for those with the various conditions are very different distributions to the healthy controls?
Well, I mean, I should say, so the actual kind of statistical results here are using linear mixed effects models that include age and sex and body mass index and median pulse transit time and heart rate, medication status, and also even the analogous interoceptive precision estimate for a control tone condition where people just tap every time they hear tone. So all of that is statistically accounted for.
And then another question. So the fact that there doesn't seem to be learning going on here, is that related to the fact that with the gastrointestinal task, it's just such an unusual thing that they're having to do that there is some learning, whereas we're used to our heart beating?
That is not actually what I think is happening. What I think the reason is because the actual interoceptive precision or just general heartbeat detection in most people at rest is just really bad. So I honestly just think that in resting conditions, the signal is just too imprecise to learn from in general. the other thing to consider is that these conditions are only a minute long, which isn't that much time to be learning anything from. In addition, people's heart rate is fairly stable, whereas in the gastrointestinal task, we were specifically varying, right, strength, and we were like had periods of time where it was on and it was off. So I think it's more just a function of the actual task and the difference in the signal strength and the kind of controllability of that signal strength.
Sure. Yeah.
So in a second sample of just healthy comparisons, we were able to replicate this effect. So it does look like it's true across healthy comparisons and when you kind of redo it in different people. Contrast, what's interesting is that the prior biases are very effectively modulated by the guessing to no guessing instruction. Or so if you go from, like, for instance, green to salmon here, right? Like, it's very clear that the prior expectation is much less biased away from feeling something in the guessing condition than the no guessing condition. But crucially, that fact doesn't differ between healthies and any of the clinical groups. So it really looks like it is something about the flexibility of the sensory precision estimate in the clinical groups, and not something about differences in prior biases or the stickiness of prior biases per se. And that's kind of important because if you read a lot of the theoretical and conceptual papers, a lot of people talk about like having bad priors for interoception as being kind of like a theoretically plausible mechanism for contributing to psychiatric disorders. And so because this task is able to separate out the kind of sensory precision weighting from the actual prior weighting, then we're able to fairly effectively show, like, look, no, it's actually not a problem with priors, or at least in the ability to adjust priors. So similar to the other task, also, if we look at correlations between the parameter estimates and things like self-reported confidence, and also self-reported feelings of intensity of the signal, they correlate with the parameters in expected directions during the breath hold, and that is also true in the confirmatory second study in the healthy comparisons. We can go back to this counting accuracy thing later if you want, but it's more of a specific interoception research point that instead of doing tapping tasks, most people just do counting tasks where they just have people pay attention and just kind of count how many heartbeats over some period of time. But you can pretty clearly show in our modeling and more generally that this counting is more or less just tracking people's prior expectations about heart rate, not really very much about the tracking of the signal per se. And it doesn't, and importantly, it doesn't relate. They don't relate at all to, like, heart rate itself, which is important. So now there was a clinical replication sample that we did as well to see if we could find the same result in new patient groups. The anxiety alone group is basically too small to look at. There was only 4 people because almost everybody with anxiety also has depression. Otherwise, the sample is generally a little bit smaller, but not unreasonable, and the kind of composition is similar. So we actually had more eating disorders, I guess. But so here, short version is that it does replicate. There's a smaller effect size, but again, it's a smaller sample slightly. So here, where this is significantly different in the breath hold compared to the other 2 in healthy comparisons, but not in any of the patient groups. Whereas again, prior biases don't differ. And if you just kind of do a linear mixed effects model transdiagnostically, where this is pink here is the exploratory sample. So the first sample in blue is the confirmatory or second sample here. Also, you can see there is a non-significant difference where the effect size is definitely less in the confirmatory sample, but the effective sample is non-significant, where the effect of healthy versus transdiagnostic clinical is significant across both samples, but only in the breath hold condition. And you can also just do kind of like a base factor comparison to say what the probability is that those 2 are actually different. And more or less what this is saying is that there's it's 3 times more likely that you'd get this data under a model where there's no difference between samples. So, and no difference again between samples in the prior beliefs. And if you combine them together, then you can combine samples together. You can, get larger ends, obviously, and you can kind of get a better estimate of the overall effect size, which I think is most impressive here. And the reason I show that is because this actually gave us a large enough overall sample size to be able to differentiate or sort of look at use. Basically what we did is we could divide people up into subgroups with a bunch of very specific disorders, right? So like in the stimulant use or in the substance use disorders patients, we could look at people who were just the people stimulant users, or just the people that were cannabis users, or just the people that were opioid users, et cetera, and just do logistic regressions to see if we could predict whether you fell into one of those groups versus whether you were a healthy comparison, or whether you fell into one of those groups versus any other group. And the main thing to see here is just that using that kind of logistic regression approach, pretty much all of the substance user groups could individually be differentiated from the healthy comparison. So it wasn't driven by a particular substance use disorder. Same story in the people with just affective disorders, whether you had major depression or generalized anxiety or PTSD, they were more or less similar to each other. One kind of interesting thing was that the social anxiety disorder could also be predicted compared to any of the other affective disorders based on the breath hold interoceptive precision. But that was kind of expected, and so we haven't really made much of it.
Can I ask about that? First of all, are those significance levels, are those all sort of Bonferroni corrected?
I don't remember, to be honest.
in the sense
I'm sure we checked that they.
Sure.
It is highly unlikely that most of those would not survive Bonferroni correction, given how significant they were uncorrected. Maybe a couple of these wouldn't, but yeah, I mean, these were certainly pitched as a more kind of like exploratory thing to do that was driven primarily by just wanting to kind of take advantage of having the samples combined. And like, to be totally honest, initially we had kind of mainly just been focusing trying to replicate the previous results. But journals often aren't too excited by pure just replication papers. So we wanted to be able to do something that was a little bit more kind of novel and kind of extending it beyond that. And so this was one way that we could do that. So these were actually not the replication stuff I showed you before actually pre-registered, like analyses designed, entirely for replication, whereas these were just kind of exploratory things that we did kind of after the fact.
So then also on the sort of on the exploratory side, is there value in looking at people who are outliers when you fit the models to them and ask about, what could be the effects of them being outliers, or why might they be outliers?
I mean, that's an interesting question. I mean, I have to think about it a bit. I mean, normally when we do, correlational analyses or anything, where our models are kind of predicting individual differences at a, in a continuous domain, then, I mean, you hope that people that are, on that any kind of individual difference predictors that we could find, would catch those sorts of relationships, unless you're talking about outliers in the sense of them being, so far outside of the regular distribution that you'd actually kind of like remove them from any kind of group level data analysis. In which case I would just worry that we wouldn't have enough data points like that to be able to say anything reliably.
But I suppose the question could be, if you look at each group and look at people on the edge of the distribution for each group and see if there's any connection between those, sort of, that is a different sort of stratification you can look at.
Yeah, I'm just trying to think. I mean, in principle, I agree. That would be interesting for sure. I'm just trying to think of if you end up with a large enough number of people that would kind of get categorized into that high-end group where you'd really have like the statistical power to do it. I mean, often, we haven't seen this here. Like in other cases, occasionally we'll get results where we see actual kind of like bimodal distributions in parameter estimates, like within a group. And in that case, we'll do things like, like k-means clustering and kind of divide people into like the high mode and the low mode, right? And then look for differences there. But again, even, I mean, anytime, right, you discretize on a continuous measure, you just kind of lose power, right? So, so it's, I typically prefer to try to keep things continuous if at all possible, but no, I mean, it's certainly interesting if it does look like in the data there is some sort of categorical difference that's being picked out, then yeah, for sure.
Elisa has a question.
Yeah, so I was wondering, in terms of your clinical groups, did you take into account current medication use and the effects on your results here, particularly with the substance use disorder? Would be more.
So there's two things. One is that in general, so medication status is a covariate that we account for in all of these analyses. And then in terms of actual kind of like substance, like preferred substance, obviously that gets kind of taken into account for here, right? Where we're looking at people who use each different substance separately. So both with respect to influences of actual psychiatric medication and with respect to substance of choice, both of those things would be accounted for, yeah.
Okay. So did you try like maybe removing all subjects that are currently on medication and just looking at the clinical populations that are not medicated at the time of your like task? Because obviously like antidepressants or stimulants or opioids would have an effect on your heart rate and your ability to like the interoception of your bodily senses, or would it have effect?
Well, I mean, I can just say, I mean, we didn't like kind of artificially separate them that way, but I mean the point is right, like I mean the in these linear mixed effects models, the coefficient, right, the estimated value for the coefficient of the medication predictor would essentially give you, right, like the estimated marginal mean for the value of people off medication versus the value for people on medication. And the point is that wasn't significantly different. Those two, like the value for that coefficient was not significant, which means that like people on medication versus off medication did not differ significantly with respect to either of the parameters.
All right, thanks.
So it's just a statistical, it's just a kind of statistically rigorous way of doing what you're saying, right?
Yeah, absolutely. I think just because I've always seen it done slightly differently with the regression models, so but I understand what you mean. Yeah, cool.
Okay, so yeah. So any summary for part 2 here, right? I mean, modeling looks like it generally supports, that people are doing something like Bayesian inference in cardiac perception, and that can differ at the individual and group level. The clinical groups, sort of surprisingly, at least based on prior theoretical work, don't show any difference in prior expectations. But they do look like they're insensitive to changes in the strength or the precision of the actual afferent signal itself. So it's a failure to kind of like adjust beliefs about signal reliability when that reliability does in fact change, was a way to think about it. So in the final third here of what I was going to show you guys, kind of draws, like I mentioned on this kind of move from just interoception alone to how interoceptive stuff influences emotional stuff. And if people are interested in kind of the more theoretical side of this, we have done some computational modeling, kind of simulation-based work looking interoception, among other things, contributes to how people infer what emotions they're feeling, and how that could influence choice. In these sorts of papers, we've shown, for example, you can have at least 7 or so different computational mechanisms within a sort of hierarchical scheme like this, where you could find differences. That could lead to differences in people's emotional awareness, and that builds on just kind of a series of prior, more kind of conceptual and theoretical papers we've done. So if people have an interest in that, they're happy to look. You're more than welcome to have a look at those. But the main point here, where it becomes most clinically relevant, is this idea of regulatory control, right? So if your brain is not very good at keeping track of the actual state of the body, then, you know, of course, it's going to have a difficult time actually regulating the state of the body, because it kind of has to have feedback about what the body's doing to see if it's regulating it effectively. And sometimes the ability to regulate it effectively actually also requires knowing something about the cause of those changes, right? So if the reason my heart rate is really high is because I'm having a heart attack, then I need to know that so I can run to the ER, right? Whereas if it's because I just exercised, then, you know, the best thing I can do is just kind of sit there and wait for myself to, to not be tired. So that requires this kind of higher level inference thing, which should in turn influence decision making. And so that's an area that we've worked a lot in the past. And so here we were specifically interested in how we might use respiration as a way to engage emotion cognition interactions, and how that might affect decision making. So here we were looking at low versus high anxiety participants in particular, and we had them complete an explore-exploit task, so a decision-making task under uncertainty with asymmetric information, while also undergoing a respiratory resistance manipulation that changed state anxiety levels. Where this respiratory-induced anxiety you can think of as being linked to panic primarily. So people breathe through this scary mask thing, and we could put different filters on the end of it that controlled how difficult it felt to breathe in. So it creates a kind of sensation of air hunger, where we can control the magnitude of that. And you can see on the right here, this just kind of shows that in both the healthy participants and in the high anxiety group, when there was the unloaded, so without the breathing resistance added, there was a difference in task anxiety between groups, but when you add the resistance or the load, then it also is effective at significantly increasing anxiety in both groups. So here there was 58 health comparisons and 61 individuals with high anxiety. The vast majority of these people also had depression, so it really should probably be called an anxious depression group. But the idea was, with this explore-exploit task, is you're kind of pitting avoidance drives against uncertainty. So you can kind of think of this as asking, okay, how much information do I need before I can be confident in the best course of action? If you're kind of too insensitive to that, if you do too little information seeking or exploit, that means you kind of jump to conclusions about the best choice too quickly. And that means that you're kind of avoiding other options that might have looked bad once or twice, but really could be better. In contrast, if you have this kind of maintained, exaggerated uncertainty that doesn't kind of reduce as it should with more observations, then you might explore too much, right? And so that's kind of the flip side of anxiety, is this kind of persistent uncertainty where you don't gain enough confidence about the best course of action, even if you do have sufficient evidence. And so question, right, like both of these things seem related to anxiety, right? Avoidance and exaggerated uncertainty. This difference between trait anxiety and state anxiety, which we can differentiate with this manipulation, with the respiratory loads. And so the task in particular is the task called the horizon task, that allows you to distinguish directed exploration, which is kind of a strategic information seeking, versus random exploration, which is essentially just your probability of choosing a low value option before attempting to maximize reward based on your total uncertainty about the best option. And you can also get measures of learning rates. In this case, we have because the model is based on a Kalman filter, you have kind of like a starting learning rate, and then you have a measure of how that learning rate changes over time as a function of changes in uncertainty. And so the task looks like this. You just have, basically there's 4 initial forced choices. So you can either choose the left choice or the right choice. And each time you choose one, a point value pops in here, and there's X's on the other side. So this would be a trial where they were forced to make 4 choices here, where there was 3 pieces of information about the reward value on that side, only 1 piece of information on that side. And then they have free choice here. And so the question is, right, do they choose the side that had the higher expected value, or do they choose the side that would be more informative, right, the side where they've only had one observation? And then we can also manipulate the horizon, right? So in some trials, they just have one free choice where gaining information wouldn't be useful. And then in other conditions, they have 6 free choices where gaining information here at the first free choice would be beneficial to maximizing reward in the long term. So you can kind of see that in Horizon 1, the information just doesn't matter to future choices. Now, the model, for sake of time, I won't go through this in too much detail, but you just have essentially a probability of choosing the right option, and that's based on the difference in the expected rewards, plus an information bonus term, right, where a higher value, the value is estimated, but it's just basically a stronger bias toward choosing the option where you've seen fewer observations. So that'd be the directed exploration. And there's also a side bias term, and then there's also a decision noise term at the bottom. And then during the forced choices, people update their expected reward values using a simple kind of risk-score reward prediction error-based update equation with a learning rate. And then the Kalman filter component, where the uncertainty is changing over time, is modeled by estimating a starting learning rate for each person and an asymptotic learning rate, which is essentially just what the learning rate would converge to if they kept playing the task indefinitely, given their underlying assumptions about the volatility in the expected reward values over time for each bandit. So here the main result is that for directed exploration, what we see is that trait anxiety is associated with increased exploration, but that state anxiety actually decreased exploration. So it is this difference where it seems kind of like trait anxiety may be kind of associated with this exaggerated uncertainty that drives people to explore more, whereas state anxiety may be more associated with this kind of like urgency where let's just kind of like jump to a conclusion. Whereas random exploration didn't show any significant effects. Interestingly, the initial learning rates showed a pattern that looked kind of like there was a general group difference. It wasn't significant, but in the asymptotic learning rates, that same pattern was statistically significant. So it does kind of look like anxious individuals are sticking closer to their kind of initial priors about the expected reward values of each bandit. So they're not updating their beliefs as much based on the observations they see in the forced choice. There's also some interesting relationships we find between directed exploration and levels of rumination, which is a depressive symptom. Basically, people just kind of internally thinking too much about how bad their lives are, as well as with a cognitive measure of non-reflectiveness. So basically, people who had higher directed exploration were also less likely to jump to conclusions about the right responses and little kind of critical thinking problems. And that builds on a prior paper that, so I should say, none of this work is published yet, but it builds on work where we, in a correlational way, had shown differences in directed exploration relating to reflective thinking and greater anxiety and depression. So summary for the final part here, increased respiratory resistance looked like it induced an interoception-based emotional state of high anxiety, successful in both groups. Greater trait anxiety looks like it led to elevated exploration, whereas greater state anxiety looked like it reduced exploration by kind of potentially creating this urgency and reducing internal reflective processes. And that greater trait anxiety was associated with slower adjustments in prior beliefs, but that was not modulated by the state anxiety manipulation. So just to conclude, kind of looked at 3 different interoceptive channels in different ways: gastrointestinal perception, cardiac perception, and respiratory-induced emotion-cognition interactions. Across tasks, one thing that seems kind of common is that psychopathology appears to be associated with an inflexibility in adjusting uncertainty estimates of various kinds. With that, I'll just say thanks to funding sources, several collaborators, and also thanks for listening.
Wonderful. Thank you so much. A really fascinating topic, quite different from what we often talk about here, but yeah, an amazing line of research. I have one quick question. So you looked at, you took into account medication status. Did you take into account meditation status, whether or not people are meditators?
Nope,
Because presumably, people who are meditators may have stronger interoceptive senses if their sort of, if their practice is about being aware of, breathing or heartbeat or whatever.
So no, we definitely don't have like a direct like measure of the degree to which people meditate. We have looked at potential relationships with different like interoceptive awareness measures that more or less like, I mean, they're self-report based, but they have to do with, the degree to which people they're kind of like related to mindfulness. Sort of things, like how much people kind of like listen to their bodies and trust their bodily signals and things like that you might think indirectly relate to the kind of like trait results of meditating or being more mindful, but we certainly haven't. We don't have anything that kind of like tracks like people's like frequency of engaging in meditation. The I should say that the interoceptive awareness, like mindfulness measures that I mentioned, those those haven't shown any relationships to any of our measures.
Okay, sure. Very good. Are there any questions from anybody? Getting some thanks there in the chat. Okay, if there are no questions for now, if you have any further questions, I can always forward them on to Ryan. But thank you so much for coming and telling us about this research. And yeah, hope we can have you back again sometime. And I will also catch you on Monday.
Sure, sounds good. Thanks
Wonderful.
again for the invite.
Pleasure.
Thanks,
Folks. Cheers. Bye-bye.